Thanks very much for this opportunity to speak. I'm recording this ahead of the conference, but I'm really looking forward to the conference itself. And thanks to the organizers for inviting me to be part of it. Today, I'm going to talk about some work that my group has been doing, prioritizing genes from genome-wide association study data. And let me start by giving some background about what that is. In a genome-wide association study, you take markers across the genome, so genetic variants, genome-wide, and one at a time, you test them for association to the phenotype that you're interested in. And the results are typically displayed in a plot that looks like this if your GWAS is successful. So here, each dot is a variant, and the y-axis tells you what the significance of association to the phenotype is of that variant. Because you typically test a million or several million variants for association to the phenotype, the correction for multiple testing is high, and so this red line here displays that threshold. And then because variants that are close to each other are often correlated with each other, if there's one variant that's associated to your phenotype, then it'll often be a member of a cluster of variants, all of which are associated to your phenotype. And so each one of these green diamonds represents a cluster of variants that's significantly associated to your phenotype. So what does it look like when you zoom in on one of these clusters? This is actually a locus from a different phenotype, but for the example, each dot here again is one genetic variant. The Y value again tells you how significantly is this variant associated to the phenotype. And you can see this cluster of variants that are all correlated to the top purple variant, and all of which are associated to the phenotype. Now, what do we want to do with this data once we have it? One thing that we might want to do is say, okay, a cluster of variants is interesting. I've identified this region of the genome that's associated to my phenotype, and I want to know what the actual variant is that drives this association. And so for this, you might want to do some kind of joint regression, maybe a sparse joint regression, but there's a lot of collinearity. Sparsity is hard. So this is an area called statistical fine mapping that there's been some really cool work in and that my group is interested in. A second thing that you might want to know is there a gene or a couple of genes that are in some sense responsible for the association at this locus? So here are the genes in this particular locus, and so what does it mean to be responsible for an association or to explain an association? One example where this is very clear is when you can narrow down to a causal variant, and that causal variant changes the protein coding sequence of one of the genes in the region. And then you might be able to conclude that probably it's that change in the protein coding sequence of the gene that leads to disease, and so in that sense the gene explains the association at the locus. But that is not a very common occurrence. That's a minority of cases, and so we often imagine that the way that the variant is contributing to disease risk is by modulating the expression of the gene or regulating the gene in some way. But as a field, our understanding of gene regulation is still incomplete. And so figuring out which genes we should be nominating and which genes we should be following up on is still a really crucial and unsolved problem for the field. So what are some ways that people have thought about this? I'll classify the most commonly used methods into two categories. One is locus-based methods. And so these are methods where that are dissecting the signal at the locus itself. So for example, such a method might first conduct statistical fine mapping to nominate causal variants that are driving the signal, and then incorporate some kind of epigenetic data, maybe variant gene interactions or some kind of predicted variant gene link, to then be able to say something like, here's a variant that's probably causal and it regulates this gene, and so this gene is probably the one that's relevant in this locus. There's a lot of different methods in this family of methods, using EQTL data, using epigenetic data, even simple methods like what's the gene that's closest to the index SNP, we would classify as locus-based methods, and what ties them together is that they're based on dissecting the signal at the locus itself. So then what's the second category? The second category is called similarity-based methods. So these are methods that now look at genome-wide patterns and that say, I think that there's probably some unifying theme, maybe there's some cell type and a lot of the relevant genes are expressed in the same cell type, or there's some pathway and I'm going to expect to see genes in this pathway show up in multiple loci. These types of methods incorporate some kind of external data like network data or expression data or pathway data. And then as a cartoon version, you might imagine saying, oh, looking genome-wide, I see that a lot of genes in these significant loci are expressed in red blood cell progenitors. Now I'm going to zoom in on my one locus that I'm interested in, and I'm going to say, oh, look, this gene is expressed in red blood cell progenitors. And that provides another kind of evidence for genes at a locus. So where did our work come in? We were kind of taking stock of the state of the art in this area, and we felt like information is being left on the table. This is a crucial, fundamental problem, but we're not actually wielding all the tools that we've got to answer it as best as we possibly can as a field. So looking first just at similarity-based gene prioritization methods, there's a lot of different types of data about genes. Mouse phenotype data, gene expression data, protein-protein interaction data. Genes have been characterized in a lot of different ways. And we wanted to build a framework that was flexible enough that we could incorporate many different data modalities. And importantly, in the future, as new data is being generated and as new types of data are being generated, that would be able to incorporate that in as flexible a framework as possible. The other type of information that we wanted to leverage was genome-wide information. So one way to think about it is, what do the genes have in common that are in these significant loci that pass this threshold? But the field now knows that there's a lot of signal in the sub-significant loci. And even if no individual locus passes significance, you can still aggregate signal across the genome and look where there's more and less evidence in aggregate. And that can be really informative about the disease. And so the first thing that we wanted to do was build a similarity-based gene prioritization method that could flexibly incorporate lots of different types of information and that also leverage genome-wide information and not just significant loci. And then the second piece, which I'll talk about less today, just for time's sake, is this idea of combining locus-based with similarity-based gene prioritization. This seems pretty straightforward, and one key difficulty here is benchmarking. It's really hard to say this new way of doing things is better than other ways of doing things, especially when what you're hoping is that you've actually taken advantage of a lot of the information that's out there, so you can't then use it for validation. So today I'm going to talk about our similarity-based gene prioritization method, polygenic priority score, or POPS. This work was led by Ellie Weeks in my group. So let me start by talking about the POPS method. We wanted to cast similarity-based gene prioritization as a regression problem. And so this is how we wanted to get the flexibility that I've been talking about. And the idea is, let's start by just creating a gene feature matrix. If we've got expression data, then let's find different ways to extract features from the expression and then create columns for each of those features. Pathways are already naturally featurized. For networks, what we do is we create a new feature for each gene consisting of all of the genes in a first degree neighborhood around that gene. Once we've put together this gene feature matrix, which is also extensible, so you can as new data becomes available, then all you have to do is turn it into columns of this matrix and then add it to the matrix. So then what would we like to do? We'd like to be able to break off one row, say this is the gene that we're interested in, and by looking only at this data, these gene features, how likely do we think it is that this gene is going to be a disease gene? So in other words, what function of this gene feature vector will give us the most information about whether a gene is likely to be a disease gene? And so in an ideal world, we would have training data. We would have lots of training data of gold standard genes that we know are disease genes or are not disease genes, and we'd use that to learn F. Of course, in practice, for the phenotypes that we're interested in, we often don't have much good training data. of this sort. And so instead, what we said was, we don't need noise-free gold standard training data. It's okay if our training data has noise in it. And so what we'd like is to have, is this gene more likely or less likely to be a disease gene? And then if we aggregate our signal over 20,000 genes, then we should still be able to have a lot of power to figure out what F is. So where are we going to get this more likely or less likely to be a disease gene? From the GWAS itself. Locus-based methods are not perfect, but they have a lot of signal. And so by using a method that gives a score to every gene in the genome, where the score is higher if the gene is more likely to be a disease gene and lower if it's less likely, that will give us a lot of noisy but informative training data that we can then use to find F. So more specifically, what do? We have a linear model. Our outcome are MAGMA scores. So this is, I should say our whole method is an extension of this method called MAGMA. And so our outcome is MAGMA scores, which are related to just the association statistics of variants that are near the gene in the genome. And our gene feature matrix is, as I described earlier, we do marginal feature selection. So we test each feature one at a time for association with Y, and then we put all of the features into X that pass some threshold. And then B is our estimand, one entry per feature, and then epsilon is our residual. And again, because of the correlation patterns of different variants in the genome, the residual has a covariance R that's not diagonal, and that was given by this MAGMA method. And so we find B, or we estimate B, using generalized least squares, which takes care of this residual covariance R, plus L2 regularization, because even though we do marginal feature selection, there's still enough features left over that L2 regularization seems to make a big difference. That gives us B hat, and then if we want to score a gene G, then we can just take the dot product between its feature vector and B hat. And one thing to note here is, as we're learning B hat, we do this in a leave one chromosome out framework, so that when we're scoring a gene, we haven't yet used the GWAS data at that locus. So that's the method. We applied it using following data. So our gene features we assembled from a number of different sources. For the pathway features, we took features that had been curated by a previous project called Depict, that had gone through and curated pathways from Go, KEGG, Reactome, and there's also a bunch of mouse phenotype gene features. We then also took the protein-protein interaction network InWeb, and as I described earlier, we created features from this network just by every gene's neighborhood is a single feature. And then for gene expression, here it wound up being a bit more of an undertaking. So Jacob and Nathan in my group downloaded and uniformly processed 77 gene expression data sets, most of which are single cell, and then applied a uniform pipeline and then extracted features in a bunch of different ways. So they took gene loadings from PCA and ICA on the whole data set. And then within each cluster, they created a feature from the average expression as well as from within cluster PCA gene loadings. And then comparisons of each cluster to the union of other clusters. So a t-statistic for differential expression, as well as a list of differentially expressed genes. And so doing this for each of the 77 gene expression data sets gave us the gene features that we're calling gene expression gene features. We then analyzed 113 phenotypes. Most of these are from the UK Biobank, and there's also publicly available summary statistics. So this included anthropometric phenotypes, as well as blood cell phenotypes and other biomarkers, cardiovascular, immune disease, psychiatric diseases, et cetera, et cetera. So once we applied our method using this data, we wanted to evaluate how are we doing. And so for similarity-based methods, there's actually some really clean ways to do this evaluation. The best, in my opinion, is the benchmarker approach. So this was developed by Rebecca Fine and Joel Hirschhorn's lab in 2019. And the question is, how much heritability is captured by SNPs near prioritized genes? And this is not circular because they use a leave one chromosome out framework. So you can say, before I even looked at this chromosome's worth of GWAS data, I'm going to prioritize the genes on the chromosome, and then I'm going to ask how much heritability is there near the genes that I prioritized. And then another metric that we used in this project was the closest gene metric. So here we're looking only at genome-wide significant loci, which limits this metric. That means that this isn't particularly good for evaluating performance for phenotypes that don't have a lot of genome-wide significant loci. But it does tell us if what we care about are these loci, then how is our performance, how similar is it to the closest gene to the index SNP? This is a way of evaluating a similarity-based method, because we know that the closest gene to the index SNP is causal more often than you would expect by chance. And that's also not information that's given to the method. So if we're often prioritizing the the closest gene, then the only way we could be doing that is by predicting the causal gene or the relevant gene at the locus more often than by chance. So here are the results for our phenotypes, benchmarker on top and closest gene on the bottom, with the phenotypes colored by category and arranged along the x-axis. And you can see that POPs is performing well from those phenotypes. So we were happy with this analysis, and it also gives us a sense for which phenotypes we think have the best performance and what the relative performances are for the different phenotypes. We then compared our method to two state-of-the-art similarity-based gene prioritization metrics, DEPICT and GWAS. And so now we've taken all the phenotypes and collapsed them into these categories. And all the way on the right, you can see that on average, POPs is outperforming DEPICT and GWAS by a good bit according to each of these metrics. And then you can also see by looking at the phenotype categories that that's the case, not just on average, but within each of these categories as well. Then we also asked these decisions that we made, can we benchmark those as well? And so one question is, we did this marginal feature selection followed by L2 regularization. Could we just have scrapped that and had one model that did L1 regularization for all of the features simultaneously? And that seems to perform less well. Then we asked, once we've done marginal feature selection, what if we do L1 regularization or no regularization instead of L2 regularization? And again, that doesn't perform as well. And then, surprisingly to me, the difference between OLS and GLS was actually pretty small. So for this particular application, it looks like, while GLS is the better method, that it's not outperforming OLS by as much as these other alternatives. So then we wanted to ask, okay, once we've run POPs, we've run it on this phenotype, and now we want to ask what features are the most important. So we can start by asking, in aggregate, we used pathway features, PPI features, gene expression features. We got performance. Is that performance mainly explained by one of those types of features? And so here is benchmarker results, again, for the different categories of phenotypes. The dark blue bar is when we run on all of the features together, and then the other three bars are if we restrict to the expression pathway or PPI features respectively. And it looks like there really is a boost for using all of the features together compared to using any of these three by itself. And then within expression, how about single cell versus bulk gene expression data? And here it looks like actually the single cell data is proving more informative on average than the bulk expression data. And that in fact, we're getting about as much out of all of the data together as we would out of just using the single cell data by itself. And then we wanted to look for an individual phenotype, which are the features that contribute the most to that phenotype. But this is complicated by the fact that a lot of our features are closely correlated. We're in a big joint model, and so the individual coefficients might be noisy for this purpose. And so we made our lives a little bit easier by clustering the features and saying, okay, we don't need to know the actual top features themselves, but we do want to know about feature clusters and which feature clusters contribute the most. So once we've clustered the features, how are we going to assess the informativeness of a particular cluster of features? Well, we'll ask how much does that cluster of features contribute to the POP scores of the prioritized genes. And so just to spell that out a little bit, the pop score for gene G is this inner product between the feature vector for gene G and the effect estimate B. And we can break this inner product as a sum across clusters of the contribution of each cluster. And so now that we've gotten, a rigorous definition of what does it mean for a cluster to contribute to a particular gene, we can then say the informativeness of this cluster is going to be the sum across all prioritized genes of the contribution of the cluster to that gene. Here are some of the results for some of the phenotypes that we looked at, where we've got the contribution on the y-axis, and some of the clusters have been labeled, and you can see that they correspond nicely to what we understand about the biology of these phenotypes. So I want to close by sharing a few examples. And one of the big pieces of this project that I'm not discussing today is how we integrated with locus-based methods and then benchmarked that integration. And I'll just say that the way that we recommend using POPs is not on its own, but in combination with a locus-based method. So to run an EQTL-based method or an epigenetics-based method, of which there are many, and then to use POPs as an extra level of evidence that can increase your confidence. So the examples that I'll present here are where we've done just that. So for coronary artery disease in this particular locus, if you apply local methods, then they actually prioritize 4 different genes. But then when you ask POPs what the top gene is in this locus, then POPs prioritizes sort 1, which is sort of a classical example of a gene that's prioritized by experimental follow-up of a non-coding signal. So that's a nice positive control. Then there's this phenotype mood swings in the UK Biobank, where people just answered on a questionnaire, does your mood tend to go up and down? And about half of the people said yes, and half of the people said no. And then they did a GWAS on that phenotype. And at this particular locus, there were 2 genes that were prioritized by local methods. And POPs prioritized DRD2, which is the receptor for most antipsychotic drugs. And then we asked, why did POPs prioritize DRD2? And the answer was because of some mouse phenotypes involving cortisol production and ion transport and anxiety. And so that I thought was an interesting example. We were able to kind of open the black box and ask, why is it that POPs is prioritizing this particular gene in this case? And then for self-reported migraine, this is another, so I'm picking out here loci where the different locus-based methods disagreed. And here POPs prioritized NGF, which is not been implicated by GWAS before, but which is known to be expressed at higher levels in the CSF of patients with chronic headache. And POPs prioritizes it because of some PPI connections, as well as mouse phenotypes, including abnormal response to pain. And then the last example that I'll present here is for bone mineral density, where the locus-based methods disagreed, picked out 6 different genes, and then POPs prioritized LRP6, which is known to be involved in bone formation, and which was implicated here because of some Wnt signaling pathways and because of PPI connections to genes involved in Wnt signaling. So we're now digging in more to these results and to characterizing these results, but I'll end here and I'll thank the members of my group who led this work, to our many collaborators, and also thanks to my funding sources and to the UK Biobank. And thanks to you for your attention.